This part explains why the chapter itself is missing from what was extracted. The source carries no chapter here. In this extraction, single neuron models, Chapter 1, is a table of contents line, a heading, an empty body, no prose to compress. The file's actual text starts at the preface and resumes at Chapter 12. What the extraction does license is position, not content. Chapter 1 opens Part 1, where the neuron is held as a single unit, before Part 2 swaps that unit for population, and Part 3 lets the couplings themselves move. Single neuron modeling is the book's base case rather than its subject. Beyond that placement, I don't know what the chapter argues and won't reconstruct it. This section explains how a detailed neuron model is built and why the whole of Chapter 2 rests on a single circuit law. A neuron is one circuit law applied twice. Current conservation across a capacitive membrane pierced by conductances, where the conductances themselves depend on the voltage they produce. That self-reference is the whole of excitability. Hodgkin and Huxley added gating variables to Kirchhoff, and nothing else. Chapter 2 installs this as the book's ground truth, then shows the same equation at finer and finer resolution. The channel zoo comes first, then synapses, modeled as channels that open on presynaptic arrival. Then the dendrite as a cable, and the cable discretized into compartments once geometry defeats analysis. Its office in the arc of the book is to make every later spiking model a reduction of something already grounded here. The surprise sits in the closing admission. Parameters multiply faster than measurements exist to fix them, so reduction is forced rather than chosen. This part explains why reducing a neuron model from 4 dimensions to 2 is the move that makes the whole chapter work. Dimension is the currency. The Hodgkin and Huxley model spends 4 coupled variables to buy biophysical fidelity, and the price it pays is geometry. Collapse the fast gating variable onto its instantaneous equilibrium, merge the slow recovery variables into 1, and 4 dimensions become 2. The whole trajectory then lives in a plane you can draw. Nullclines, fixed points, and their intersections replace numerical integration as the explanation of why a neuron fires. Threshold, refractoriness, and repetitive firing appear as phase plane structure rather than as simulation output. This chapter is the pivot of the book's arc. It is the last stop where the membrane is still a mechanism, and the first where firing is a geometric event. This part explains how to describe a neuron without tracking its internal state. Drop the state variables and keep only the spike times. The membrane potential becomes a sum of 2 kernels, one for the refractory aftermath of the neuron's own last spike, one per presynaptic spike, and a spike fires when that sum crosses threshold. This is the spike response model. Leaky integrate, and fire is its special case with exponential kernels. The move is a change of coordinates from differential equations in conductances, the subject of chapters 2 and 3, to an integral operator on firing history. So the neuron is a linear filter followed by a threshold. Tractability at population scale is what that buys. Anything not expressible as a response to past spikes is what it forfeits. This section explains what noise does to a spiking neuron model. A deterministic threshold makes a neuron a function. Noise converts it into a point process, and that conversion is the chapter's whole work. There are 2 structurally distinct entrances. Randomness can be placed at the threshold, which is escape noise, an instantaneous firing intensity set by the momentary distance to threshold. or randomness can be placed in the input, which is diffusive noise, stochastic spike arrival driving the membrane potential as a random walk. Both entrances return the same object, an interval distribution with its survivor function and its hazard rate. The primitive is this: a spike time becomes a quantity you assign probability to rather than a quantity you solve for. That probability is precisely the currency the population equations of Part 2 spend. This section explains how a whole population of neurons collapses into a single quantity, and what that quantity obeys. One state variable replaces the n neurons of a population, A of t, the fraction of the population firing per unit time. It is an ensemble average taken at an instant, where the familiar firing rate is an average taken over time. Make the network homogeneous and infinite, and that single variable closes on itself. The activity's own past feeds back as the input current, driving its future. The chapter's payoff is a duality. Track the density of neurons across internal state, which gives partial differential equations and a microscopic picture, or track A of t through input-dependent interval distributions, which gives one integral equation and a macroscopic picture. The two coincide. Everything Part 2 does afterwards runs on that equation. Transients, oscillations, spatial patterns. This section explains why a population of neurons answers an input faster than any single membrane can charge. The membrane time constant does not set how fast a population answers. Linearizing the population equation of Chapter 6 around asynchronous firing shows that the response to an abrupt input is carried by the neurons already sitting infinitesimally below threshold. What moves is a flux across the threshold, a crossing of neurons already poised there, and the charging curve of a single neurons already poised there, and the charging curve of a single membrane is the wrong quantity to look at. With low or slow noise, the activity follows H prime, the derivative of the input potential, so the population reacts with no integration delay at all. Strong, fast noise smears the crossing, and under that condition, the ensemble finally behaves like a filter. Ergodicity closes the argument. N independent neurons measured in one trial equal one neuron measured over n trials. That equivalence is what makes peristimulus, time histogram, and reverse correlation experiments direct measurements of this transfer function. This section explains why the asynchronous firing assumed in the previous two chapters is generically false, and what replaces it. Chapters 6 and 7 assumed asynchronous firing. This chapter audits that assumption and finds it generically false. In a homogeneous network at low noise, the recurrent coupling destabilizes its own fixed point, and the population falls into coherent oscillation, cluster states, or sparse network irregularity locked to a global rhythm. The entire stability question then collapses onto one local test, the locking theorem. A synchronous oscillation is asymptotically stable exactly when spikes are triggered during the rising phase of the summed postsynaptic potential, a global dynamical property read off the sign of one derivative at one instant. The sign of the coupling does not matter. Excitation and inhibition both lock. Asynchrony survives only where noise and axonal delay conspire to sustain it. This section explains what happens when a population of neurons is given a spatial coordinate. Drop the assumption that every neuron sees every other one identically, and the population equations of Chapter 6 acquire a coordinate. Coupling becomes a function of distance alone, and the network becomes a continuous field. That single move is what the chapter is for. It converts a rate dynamics in time into a dynamics in space and time, where the shape of the interaction kernel selects what the tissue does, where the shape of the interaction kernel selects what the tissue does. Mexican hat coupling near excitation and far inhibition destabilizes the homogeneous state, and the wavelength of the instability picks the pattern. Stationary activity bumps, traveling waves, orientation tuning that persists after the stimulus that seeded it. Structure in the connectivity becomes structure in the activity. A spatially blind population can hold no place. This section explains how Hebb's postulate becomes a formal model and why that formalization generates a whole family of learning rules. Hebb's postulate is a sentence about coincidence. The chapter turns it into a local rule, a weight change written only in quantities. The synapse itself can see presynaptic input and postsynaptic state at the moment of change. Locality is what makes it a model rather than a slogan, and it generates the whole taxonomy. Expand the weight change as a Taylor series in presynaptic and postsynaptic rates, and every classical rule falls out as one choice of coefficients: pure correlation, decay, sliding thresholds. Replace rates with spikes, and a coefficient becomes a learning window over pre-post timing. Plasticity enters part 3 as a measurable coincidence detector carrying free parameters a slice experiment can pin down. This section explains what a plasticity rule computes once you stop asking how it changes a synapse. Chapter 10 hands you the rule itself. This one asks what quantity the rule is estimating. Average, a local Hebbian rule over the input ensemble, and the weight vector obeys a linear differential equation whose generator is the input correlation matrix. Learning therefore becomes an eigenvalue problem, and the weight vector rotates into the principal component. That is the skeleton, and everything else arrives as a correction to it. 2 corrections matter. Unbounded growth demands normalization. And, in the spiking version, the correlation matrix becomes the learning window convolved with spike train correlations, with rate learning surviving as its slow limit. The chapter's place in the arc, it converts a mechanism into a statistical operator, so plasticity becomes predictable rather than merely defined. This section explains how a single timing asymmetry in synaptic plasticity generates both learning and the code a network can carry. One asymmetry does all the work. A learning window, written W of S, that rewards presynaptic spikes arriving before the postsynaptic one, and punishes those arriving after, is a causality filter. It keeps exactly the synapses that could have caused the spike. Every phenomenon in the chapter is that filter applied to a different substrate. Place fields drift backwards along the rat's track. Jittery inputs are pruned in favor of precise ones. Delay lines are selected until a sequence replays at its learnt speed. The electric fish subtracts its own predicted image. Coding and learning stop being separate questions here. The code a network can carry is whatever its learning window selects for. This part explains how a timing rule inside a single synapse turns into a law of behavior. Asymmetry in time becomes prediction. The learning window, a function of the difference between the postsynaptic and presynaptic firing times, rewards a synapse for firing before the postsynaptic spike and punishes it for firing after. Iterate that over a repeated input sequence, and the surviving strong synapses are the ones that came first, so threshold is crossed ever earlier. The response walks backward along the causal chain until it sits on the earliest reliable predictor. Across 100 trials, the latency moves from 31 milliseconds to 5 milliseconds. The hippocampal place field center slides against the rat's direction of travel, and Pavlov's bell takes the response over from the food. One sign convention in a synaptic rule, one sign convention in a synaptic rule, one behavioral law. The window's width, 50 to 100 milliseconds, is the ceiling. Seconds. Long conditioning needs extra machinery. This section explains how one learning rule produces two opposite outcomes, depending on a single number. The window is asymmetric. The two selections it can perform are opposites, and what chooses between them is a scalar, the firing threshold. Section 12.1 had this window favor the earliest synapses. Here, the same window, with the same parameters, favors the most precisely synapses. With a low threshold, a few early jittery spikes fire the neuron before the punctual group arrives, so precision gets depressed. With a high threshold, almost nothing fires, and the non-Hebbian presynaptic term saturates every weight indiscriminately. Only in between does the sloppy group fail to trigger while the precise group reliably does. Heb then strengthens exactly those precise synapses, and coding efficiency peaks near a threshold theta of about 0.25. Selectivity is a property of the operating point rather than of the rule itself. This section explains how the model is built and what its construction rules out. Strip every asymmetry from a synapse population except one, the width of the spike volley each afferent delivers. Everything else is held identical: the kernels, the normalization, the mean spike count per presynaptic cell. Whatever weight structure then emerges is caused by temporal jitter and by nothing else. The model is a controlled experiment written as dynamics. Two soft edges carry it. The threshold is an escape rate rather than a cut, so beta grades reliability, meaning how fast firing follows a threshold crossing. The learning window is exponentially asymmetric, potentiating before, depressing after. Weight-dependent amplitudes do the stabilizing work. Potentiation is scaled by 1 minus W, depression by W. Together with the non-Hebbian drift terms, these pin the neuron at its operating point so nothing runs away. This part explains why the firing time distribution takes the form it does, and what plasticity actually reads off it. Learning needs a correlation between presynaptic and postsynaptic activity, but the nonlinearity of the firing threshold forbids computing one exactly. Tractability is therefore bought twice by two separate approximations stacked in sequence. Dilution comes first. With many presynaptic cells, any single input contributes negligibly to the output, and the joint density factorizes into a product of marginals. Then the doubly stochastic output, a random membrane potential feeding a random emission, collapses to an inhomogeneous Poisson process. Its intensity is the escape rate averaged over the input ensemble. That averaging discards refractoriness, which returns as a survivor factor. Attend only to the first spike, weight the instantaneous rate by the probability that no spike came earlier. What plasticity actually sees is a first passage density. This section explains why synaptic weights stop changing under multiplicative soft bounds and what the resting value encodes. Multiplicative soft bounds turn plasticity into a fixed point. Every potentiating term is scaled by 1 minus Wij, and every depressing term is scaled by Wij. So under a repeated stimulus, the two contributions balance and the weight stops moving. Where it stops is a function of firing time statistics alone, the summed jitter sigma and the mean pre-to-post lag S. The stationary weight therefore reads out the temporal precision of a synapse's input rather than its activity level. At small sigma, the equilibrium profile reproduces the learning window itself, saturated near 1 for before post, near 0 for the reverse ordering. As sigma grows, the Hebbian contribution averages away, and the non-Hebbian terms alone fix an intermediate value. What survives at equilibrium is precision. This part explains why the firing threshold, and not the learning rule, is what selects which presynaptic population wins. Set the threshold and you have already chosen the survivor. The learning window never changes. What changes is who gets to fire the neuron first, and the threshold alone decides that. Take the threshold low, and a few early spikes suffice. Early means jittery, so the sloppy group is potentiated while the precise group arrives after the spike and decays. Take the threshold high and almost nothing fires. The non-Hebbian presynaptic term then potentiates every synapse indiscriminately. Between the two sits a narrow band at roughly 0.25 of the maximal potential, where scattered early spikes cannot cross, but the synchronous group can. Precision is consistently rewarded there, and postsynaptic jitter collapses. Selectivity is non-monotone in threshold, and coding efficiency peaks inside that band. This part explains how a network learns a sequence in time. An asymmetric learning window converts temporal order into synaptic structure. Fire J before I, and the synapse from J to I grows, so replaying J drags I behind it. The sequence becomes a synfire chain, and the trained network reruns it with the external teacher removed. The subtlety is speed. Timing survives only if the window selects, among the many axonal delays between the same pair, the one that could have caused the postsynaptic spike. Its maximum sits inside the rise of the EPSP, the causality condition of Hebbian learning. Memory of time is stored as a choice of delay line. Here is the chapter's turn. The mechanism works, and the code still probably isn't cortexes. Past about 20 patterns in 1,000 neurons, the dynamics collapse. This section explains how a neuron learns to fire only at surprise. A neuron that fires only at surprise is one whose synapses have learned to subtract its own prediction. Reverse the sign of the STDP window so that presynaptic slightly before postsynaptic now depresses, and Hebbian reinforcement of temporal structure becomes its cancellation. Whatever component of the input is predictable from the delay line basis gets driven out of the input is predictable from the delay line basis gets driven out of the T. The marmorid ganglion cell is the anchor. Delays spanning 0 to 100 milliseconds after each electric organ discharge span the space of predictable waveforms, and anti-Hebbian plasticity performs the least squares projection onto that space. The tell is the negative after-image. Remove the trained stimulus and the learned subtrahend stands exposed. Novelty detection needs no novelty detector, only a learning rule whose fixed point is prediction error. This section explains how a weakly electric fish builds perception by subtracting its own predictions. A fish that illuminates the world with its own electric discharge mostly sees itself. Each pulse returns a field dominated by the fish's body and its fixed surroundings, so an object arrives as a small deviation on a large predictable background. Marmorid medium ganglion cells receive two streams. One is a delay line fan carrying when the electric organ fired, spread across 0 to 100 milliseconds. The other is the electroreceptor image itself. The cells learn to make the first stream cancel the second. Only the unpredictable survives. What results is a novelty detector whose selectivity is manufactured by subtraction. The signature of that manufacture is the negative after-image. Withdraw the trained stimulus, and the cell reports its inverse, the stored expectation now canceling nothing. Perception here is the residual of a prediction, and the prediction lives in synaptic weights. This section explains how a neural circuit learns to erase what it can predict by running Hebbian learning backwards. Flip the sign of the learning window, and a structure detector becomes a structure eraser. The Mormira ganglion cell receives its own electric organ discharge through 150 delay lines, moment by moment by Mayans, a temporal basis, together with an anti-Hebbian rule. Under that rule, a presynaptic spike arriving slightly before the back, propagating broad spike depresses the synapse. The rule tunes those weights until their weighted sum cancels the sensory image moment by moment. The fixed point is a mean rate. Whatever repeats is subtracted, and only the unpredictable survives. This gives novelty detection built out of cancellation rather than comparison. Remove the stimulus after adaptation, and a negative after image appears. The expectation lived in the weights, not in a gate. This section explains how the barn owl builds a code finer than the parts that carry it. The owl resolves intraoral time differences down to a few microseconds using synapses whose time constants are 100 microseconds. The code is sharper than its own components. Coincidence detection buys that precision, a volley of phase-locked, excitatory postsynaptic potentials must cross threshold on its rising flank, so output timing inherits the sharpness of arrival rather than the width of the membrane. Jitter falls from 40 microseconds to 25 microseconds across a single relay. This works only when the delay lines share a preferred phase, and that alignment cannot be genetic in a head that grows. Here is the chapter's closure. The same spike time window that reads timing also selects the lines that carry it, with survivors differing by whole stimulus periods. Coding and plasticity, one loop. This section explains how the owl's auditory pathway converts a difference in arrival time into a location in space. 5 microseconds of interaural delay carry the owl's 1 to 2 degree localization, yet each spike arrives with about 40 microseconds of jitter. The quantity to be resolved sits an order of magnitude beneath the noise of every element resolving it. Jeffress made his move in 1948, and Carr and Konishi found it in the nucleus laminaris 4 decades later. It converts the time difference into a place, signals race inward from both ears along transmission lines, and the coincidence detector that fires is wherever the two wavefronts happen to meet. Time becomes position. Precision that no single neuron possesses becomes the identity of a unit in a population. Cochlear frequency separation makes each channel carry a phase. A phase is periodic, hence ambiguous, and that ambiguity is resolved further up by combining channels, not here. This part explains how a neuron locks its output spike to the phase of its input. A threshold can only be crossed from below, so the output spike is pinned to the rising flank of the last volley. That single geometric fact carries the whole subsection, phase in, phase out, with no mechanism beyond linear integration and a fast membrane, one whose time constant is 0.1 milliseconds, bought physiologically by an outward rectifier. Convergence then buys precision. 154 noisy lines averaged, sharpen an input, jitter of 40 microseconds into an output of 25 microseconds. This is the square root of n story told in the time domain rather than the amplitude 1. The condition is that the summed potential must oscillate. Draw the delays from a uniform phase distribution, and every line stays perfectly phase locked while their sum goes flat. Coherence is a relation among lines, invisible in any one of them. This part explains how an owl builds its own microsecond delay lines without a blueprint. An owl's head grows through development. A genome cannot pre-specify microsecond wiring, and there is no supervisor to hand out delays. So the coincidence detector in the nucleus laminaris must grow its own delays from spike timing, the only signal available locally. Gerstner and Kissler's move is a learning window with a negative integral, plus a small, non-Hebbian term that pins the postsynaptic rate. That stabilization turns learning into competition among the roughly 600 converging lines. Synapses, whose spikes land on the rising phase, saturate. The rest decay to zero. Roughly 150 survive, agreeing in delay modulo the period t, because phase is all the rule can see. The Jeffress array is distilled out of the very signal it will later measure.